Transcription begins with an enzyme called RNA polymerase. RNA polymerase attaches itself to a promoter on the DNA strand. As it does this, it reads the complementary base pairs and copies them onto the mRNA strand. Once it reaches a stop codon, the mRNA dissociates from the DNA molecule. And this mRNA strand is now called a pre-mRNA transcript. Before the pre-mRNA molecule can leave the nucleus, it must undergo editing. First, a guanine cap at the 5' end is added, and so is a poly-A tail at the 3' end. Then, introns, which are non-protein coding regions, are removed by spliceosomes. After this, the mRNA molecule is ready to leave the nucleus through the nuclear pores. Initiation is the step that brings all the translation components together. Proteins called initiation factors are required to assemble the small ribosomal unit, the mRNA initiator, tRNA, and the large ribosomal unit for the start of protein synthesis. Elongation is the stage during protein synthesis when a polypeptide increases in length one amino acid at a time. In addition to the necessary tRNAs, elongation requires elongation factors, which facilitate the binding of tRNA anticodons to mRNA codons within a ribosome. As the polypeptide chain is being built, translocation occurs. The ribosome moves forward and the peptide-bearing tRNA is now in the P site of the ribosome. The spent tRNA, now at the E site, exits the ribosome. A new codon is now exposed at the A site and is ready to receive another tRNA. This goes on until the ribosome reaches a stop codon. Termination is the final step in protein synthesis. During termination, the polypeptide and the assembled components that carried out protein synthesis are separated from one another until they are ready to be used again. The polypeptide chain, now dissociated, is free to take its final three-dimensional shape. It may also combine with other peptide chains to form a fully functional protein.